Empezamos. Muy buenas tardes. Soy Emilio Bouza, presidente del Consejo Científico de la Fundación.
Good afternoon. I'm Emilio Bouza, President of the Scientific Council of the Foundation. I'd like to welcome you and thank you for your presence here today. And I would also like to explain, in case you haven't attended these special sessions, such as today's, I'd like to go back in history. 18 years ago, Springer and the Foundation reached an agreement according to which, on a yearly basis, we arrange a leading topic for a joint session. Several proposals, and what we have chosen this year cannot be more relevant, of course, and very cutting edge. When I was looking at the title of omics, of single cell and the human cellular atlas, I believe that we are witnessing a landmark, a true landmark in the history of medicine. When you go back in history, concerning the greatest landmarks in the history of medicine, it is certainly very much related. In the 1600s, Andres Vesaliu in Padua University was the first to describe human anatomy, the Humanis Corpori Fabrica, in the colloquial language, the Fabrica, and how the human body was manufactured and it's structured. A century went by and somebody discovered the microscopic world. And there are things that cannot be seen at the naked eye, but with the application of different lenses can be visualized. Things that we just couldn't see at first sight. And this is not so well appreciated, actually. The microscope was evolving, and then it was used as a leisure element for the rich until the 19th century, until somebody realizes that this is a key instrument for scientific research. We have the leading researcher who realizes that in human tissues there are some cells that were similar to honeycomb cells, and the cells that are seen in the vegetable tissues. And that's the true cellular research beginning. Tissues were composed of cells. And then the end of the 19th century, there is a great revolution in our field, which is closer to my field, microbiology, and people discover microbes associated to infection. But they were working with everything. Altogether is chaotic, like Pasteur did, right? And then finally, Roberto Koch was the one to, for the first time, with the aid of health, to manufacture a solid medium for culture in order to isolate the colony-forming unit that produces a colony with a single microorganism. He was the first. During the same years, there was another great breakthrough. Human cells may be harvested and cultured. And therefore, we are talking about Rox Harrison's research, and we may have cellular culture. 20th century entails the discovery of DNA and its structure, but omics have been working with human samples that represent the set of cells of diverse origin, as well as many other substances. Nowadays, we can work on something that a single cell and its progeny produces, and that is a great landmark to be added to this list. If Antonio Koch and the other scientists that I mentioned could be sitting here today, they would be so happy and very pleasantly surprised to listen to today's speakers. I will not tire you any further. I would just like to mention that this joint activity with Springer Nature, all the Nature journals, is a wonderful activity. We're very happy. We hope we will have continuity in the next years. And I'll just like to call on Soledad Santos, the director of Springer Cell Europe, to start moderating the session. She will give the floor to Erika Pastrana later, and we will have an incredibly didactic session. I'm sure we'll all be learning A great deal. Thank you very much, and Soledad, you have the floor. Thank you. Thank you very much, Emilio, and thank you very much, everybody, for being here today at this 18th meeting. 1 year more, we meet together to listen to cellular biology advances by the pioneers in the human cell atlas and omics. And our meeting wouldn't be possible without the excellent cooperation between Springer Nature and the Ramon Areces Foundation, almost 20 years. Now we are looking for new relationships and new research networks. Such a long-standing collaboration can only be explained because of this very strong commitment by both Springer Nature and Ramon Areces Foundation have for the dissemination of high-quality science. Both of us are driven by the spirit of scientific dissemination, ensuring the quality of the science we explain, always playing with the plain objective of the sustainable development of society. We want to thank the director of the foundation, Mr. Raimundo Fernández Torra, with this collaboration. Thank you very much. We collaborate with Springer Nature and the scientific committee of the foundation, presented by Professor Boza, who was addressing you, and Jose Maria Medina, who are the perfect recipe to choose the best topics. They have a great intuition and knowledge, and so we talk about very much cutting-edge topics. Even society doesn't know about them. And after all, we want to thank Mr. Azcona for the trust that he has placed upon us for many years, and because we have learned so much from him, and it's a great privilege to learn such a great master. The project Human Cellular Atlas started in 2016 with an international spirit in trying to have an atlas of all human cell types, because if we knew better about each cell in each tissue, we would learn much better about how the body works, both physiologically and pathologically. And this is sustained by key values of scientific progress, which is the networking collaboration with researchers all over the world, standardized scientific methods to be applied so as to be able to compare the results, and the commitment to allow free access to the data published by anybody, and ensuring diversity and representativity of the samples employed. If we were to understand the human body as a great symphony where hundreds of musicians play different melodies in harmony, HSCA is deciphering the kind of music each musician is to be played, differentiating what is it that makes all cells to act in our body in a coordinated and harmonic fashion. From the beginning in 2016, there's been more than 300 researchers of 102 countries publishing 250 articles in peer-reviewed magazines, and as the output, Nature published in 2024 the first collection of what was the first draft of the Human Atlas. And our commitment goes further. Right now, we have a collection focused on Cancer Cellular Atlas, collecting all different articles to characterize the composition, heterogeneity, and the response to different treatments of the tumor cells. We urge you to read this fascinating collection and send your contributions if you wish to participate. However, we wouldn't be here today talking about the Human Atlas if it weren't for the advances in the last years in the single-cell omics, studying genomics, transcriptomics, and proteomics at the single-cell level, and together with spatial advances have placed the location of each cell and its dynamic evolution in time. We have 4 experts today who will bring us closest to the great results and the multiple possibilities opened by this project, joining the best techniques with the best minds in order to advance to a more precision medicine. Thank you very much. Yanai Ital, Roser Vento Tormo, Maria Colometache, and Holger Hine, and the Vice President of the Nature Journal for participating in this event. Thank you very much, everybody, for your attendance, and I hope you enjoy the presentations and the posterior debate.
Thank you very much, Emilio. Thank you very much, everybody, for your attention. And I hope you enjoy the presentations and the posterior debate. Thank you very much, Emilio. Thank you very much, everybody, for your attention. And the posterior debate. Thank you very much, Emilio. Thank you very much, everybody, for your attention. And the posterior debate. Thank you very much, everybody, for your attention. And Buenas noches a todos.
Good evening, ladies, gentlemen. Thank you very much for coming here in Madrid. It was a wonderful day outside, and it's a great pleasure to be introducing today's speakers. We've already set the background on tonight's issue. I'd just like to emphasize that I urge you tonight to participate. If you're a biologist, as I am, many of us are, studying the cellular function in biology, we are now sitting here with open minds. Please try to welcome a new view of cellular functions, a detailed and dynamic vision of what a cell is. Cells 10 years ago, even 5-7 years ago, were so much more limited. But now we have these current techniques that have allowed us to analyze the cells at a very detailed level in the spatial and temporal and genomic level, are opening the doors to redefine what is a cell right now, how they change, evolve, relate to each other, age, and sometimes degenerate, of course, in cancer. So I urge you to keep an open mind perspective, and I'm sure the four speakers today will highlight a new approach of knowledge of the human body thanks to the study of the cell. My colleagues have already introduced this very important, relevant project of the Human Cell Atlas, and we're here to celebrate the international networking that facilitates projects as the HCA. It's wonderful to see that science doesn't advance isolated silo by silo, laboratory by laboratory. No, we are certainly a spectacular time where scientists are joining as musicians of a single orchestra, producing this joint symphony. The Human Cell Atlas represents the collaboration of many international laboratories with a common objective: decipher the detailed map of the human cell maps, the atlas. And it's very gratifying to see what the scientific community can do when they join a common goal, which is so very relevant. So without any further ado, I would like to introduce the first of today's speakers, Professor Yanai. Professor Yanai is a professor at the Grossman Medical School in New York at genetic systems. He deals with computational models, philosophy. He has a doctorate in science as well, and his laboratory has been a pioneer in the single-cell sequencing method that has revolutionized the way we study interaction, progression of cancer, host pathogen interactions, embryonic development, all through global gene expression. Dr. Yanai is going to be surprising. He's going to talk about the evolution of programs of genetic evolution, plasticity, and also he is co-author of the general dissemination book, The Society of Genes, as exploring the interdependent relationship, and the Knight Science Institute, of which he is a founder. That is a fascinating project to combine creativity and scientific spirit. So, please, Professor Yanai, you have the floor.
Okay, how is your English? Thank you, Erica. Wow, it's really a pleasure to be here. I love Madrid, and really today has been like a first-class tour of the city, and it's been a lot of fun. So I'm really looking forward to talking to you today about cells and their state. So, okay, what I want to tell you about is what we're learning in terms of principles. What have we learned about cells with the new technology? What mysteries we still have? And also, I think, the process that underlies discovery. So, to get started, what I want to tell you is that, of course, we've been studying cells for a very long time. We have had the microscope as the main tool. And for many years, we've studied what cells do by just looking at them. And we see, what do they do? What do they look like? And that was our best characterization of them. And of course, one of your own countrymen, Santiago Ramón y Cajal, is a legend in science in general, and specifically in neuroscience, for having first characterized neurons and classifying the different branching types and the spatial organizations. And so for a very long time, we've had this heritage of trying to classify cells. Well, what's happened over the last couple of decades is that we have a new technology. In a sense, it's like a new microscope. It allows you to see things that we just haven't been able to see before, and it's really exciting. In my lab, we were very fortunate that we were one of the first ones to set up a method for this tool called single-cell RNA-seq. And basically what this tool does is that, I have to use my hands, instead of just looking at the cell, you get a high-dimensional profile for every cell. In a sense, it's a vector of 20,000 numbers, one way to look at it. 20,000 is because there's 20,000 different genes in every cell, and those genes could be off, so then it would be a 0. Or that particular next gene could be on, so then it may be 10, or maybe it's expressed higher at 100 copies of mRNAs per cell. And so you have this vector that you get for every single individual cell. And then when you have that, you can now do all kinds of analyses. And it didn't have to be this way, that by just studying the mRNA, you could see something. But it just so happens that the way nature is organized, if you count mRNAs, in a sense, that's the first phenotype of the genome, then you have a really good representation of what that cell is doing. It didn't have to be this way, but it just is. And so what we find is that when we compare these vectors, right, you have one vector of 20,000 for one cell, you could now compare it to another. And then, it's really hard for us to see more than 3 dimensions. We're just human. So we use a dimensional reductional algorithm. And then here, what you see is that every point indicates a cell, and you can see their relative organization. And what we see is that there are clusters, right? You see that there's islands. And it turns out that what those islands are cell types. Something that didn't have to be. And it still kind of shocks me that that's how nature is organized. No matter how many times I've seen it, it still shocks me. But we see that what you may call the first law of transcriptomics, which is that different clusters correspond to different cell types. Okay? And that means that you could almost now have a program for what is each cell type. You have a molecular fingerprint. Ramón y Cajal looked at the cell, the image. Here, we can look at what the cell is molecularly. What are the genes? And we can say, that gives you like a kind of circuit. And for every cell type, we can have a circuit. We could say, what is a delta cell? A delta cell is this kind of circuit. What is, I can still stand here, right? Oh, good. What is that kind of cell type? It's a different kind of circuitry. And that's really interesting, right? That there's different circuitries for every cell type. However, you may then fall into a kind of Platonic trap. Remember, Plato saw the world as having ideal forms. So you can each imagine a triangle. Like if you close your eyes, you can imagine a perfect triangle. But then if I asked you to draw it, it would be an imperfect representation. And I think in science, we kind of fell into a Platonic trap where when we looked at the transcriptome, so that's that vector for every cell, you could imagine, it's a noisy representation of an ideal state. Really, what we have been finding lately is that there is no ideal state. Really, there are a lot of intermediate states, and cells are able to transition. And nowhere is that more obvious than in development, right? Because each of you, even though it's so astounding to believe, but you have to believe me, that each of you was at one point one cell, right? And then you were 2 cells. It had to be this way. There's no way you could argue with this fact. And so what happened? How did this diversification of cell types arise? Well, there had to be transitions. There had to be changes, right? And so we can, with this technology, see it before our eyes. Let's say you take a testes sample. This might be a sensitive issue with some people, but we could take a testes sample, and again, we see the action of the first law, which is there's different clusters. So you see the Sertoli cells, the Leydig cells, those are different cell types. But then you see one cluster that goes around. You see it has a shape, like it's not a simple cluster. There's some structure to that cluster. Well, what do you see there? You really see cell state transitions. You see changes. And these, it turns out, is the differentiation of spermatocytes into sperm. And so every dot there is a cell. We measured the transcriptome, and they're all lining up like soldiers representing the differentiation process of turning from one thing into another. And they're all just unfolding the process for us before our eyes. It's really amazing. So, okay, you can say this is the second law of RNA-seq, right? That not only the clusters represent cell types, but then there's structure inside of a cluster that represents different states. Okay, this is very good. But now there's a third thing that we find ourselves wrestling with, which is that in many cases, there are new states that appear. You can have a change in the environment, and suddenly cells would now migrate to a new state. And that's really interesting. Like, how would that happen? How can a new state arise? And so this is something that we don't really have an answer to. I think it's the future for us scientists to tackle this question. I think it's sometimes very misleading when we scientists talk about science. It's as though everything's already been discovered, but it's not true. There are so many things we don't know, and all one needs to do to prove that we don't know things is to point to a hospital where you see people are dying. So that shows that we don't understand things, that there's things that maybe we could treat better. And so one thing we don't understand is how cells transition to new states. And I want to tell you quickly about an experiment that we did in my lab. So in cancer, when we go to the hospital and we see that people die of cancer, like, why do they die of cancer? Do we not have treatments? No, we do have treatments. We give drugs, the drugs work, and then suddenly the drugs stop working, right? The way the field talks about it is that the cells are in a sensitive state, the drugs work, and then they transition into a resistant state. Suddenly the drugs no longer work. It was always thought that it was because of mutations that arise. But lately, and in particular, thanks to this new technology of single-cell RNA-seq, we could see that there's an additional layer to this. There are cell state transitions. And so I want to tell you about the experiment that we did. We took cells, you can grow cells in a dish in the lab, and they grew up. And you see they grew up in 6 days. Then what we did is we added a drug to this cell. We added a drug of, you see where it says 1 up there? We give a little bit of a drug. And now the cells grow, but not as fast. It takes 10 days, because that's a cumulative number, so 6 plus 10 is 16. But then they adapt to that 1 micromolar. Then what we did is we increased the dose from 1 to 2.5. Again, the cells take some time, but they do adapt. And then we double the dose to 5, to 10, to 20, to 40, to 80, to 160, to 320. Every time the cells adapted, we doubled the dose. And then we asked, what do you see? What happens? The way we can look at this is we ask, what is the cell viability? So you see here, you take the cells that are drug naive. And if you give no dose, 0, they're 100% alive. But as you add the dose, the cells die. That's what you expect, because these are cells that have never seen the drug. What would happen if we now do the same analysis for those 9 lines? Well, if there is just a simple to resistant switch, they should all look like this, right? This is fake data. I'm just hypothesizing. Maybe it looks like this. what do you think it would look like? That's why we do science. We do the experiment and we let nature tell us. This is what we actually saw. For each of the 9 lines, there's a shift to the right. So you see, the ones that grew in the presence of the 1 micromolar dose, they shifted a little bit to the right. And then when we added the dose to 2.5, it shifted. Every time we challenged the cells with a higher dose, the cells, they took time, but they adapted to that dose. So there is no sensitive to resistant switch. There is no resistant line. There are resistant lines. There's different, right? There's multiple ways. And so we were interested to see what happens. Well, with this technology, this new microscope, as I mentioned, you can do the transcriptomics for each line, you see for each of the 10, and you can see that there's different subpopulations that arise. When you take the average of each subpopulation and compare averages, what would it look like? What do you think? I mean, what's happening here? What we saw is that if you, for example, take these 4 subpopulations and take the average of them, it's these 4. And when we do a correlation, red is high correlation, so very similar. Blue is very low correlation, very different. You can see that these 4 are the same. And what's interesting is that there's actually not just one like resistant state, but there are 5 resistant states. You see this clustering? So as we added the drug, this technology allows us to like look under the hood. You know, like let's say the car is broken. You say, oh, the car is broken, what can I do? But with this technology, we can open the hood, look inside, and when we look at the genes, we see that inside there are different states. And you can tell it kind of like a movie where, okay, the drug naive, that's the control. They're in one particular state. As soon as we added the drug, all the cells were in state 1. When we increased the drug again, there was a new state that was born. A new state. Where did it come from? This is called state 2. That state 2 grew in frequency. By the next time point, it actually made extinct state 1. You see this yellow disappears here, but then 2 new states arise. They get very popular. They kick out state 2. They become very popular, and then a new state, state 5, emerges. So new states keeps popping up, and it's a real mystery of like how it's doing it. And so what have I told you is that with this research, which I'm just showing you a little taste of, instead of a sensitive to a resistant switch, we see lots of cell state transitions, different states. And it's a real mystery for us how the cell is doing. I want to tell you in the few minutes that I have left what my pet theory is. You know, every scientist has like a pet theory for what's going on, like the best guess that we don't have all the evidence for, but it's what is happening live in the lab right now. Well, so here's my idea of what's happening. First of all, it's not what we've been taught. So for a long time, we've been thinking about what a cell is doing as executing programs. We have been looking at, and I'm just as guilty as anyone in the field for doing this, we've drawn circuit diagrams to indicate how every gene is regulating every gene. So you see, every line here is indicating a gene, and you can see that one gene can like turn on other genes, and it's like a circuit diagram of what's happening inside the cell. There's programs, and with a particular input, there could be a particular output, and some people say that what we should do is elucidate the gene regulatory networks that are everywhere in the cell. So imagine, like, what are you? Like, how did you go from one cell to many cells? Maybe it's the execution of a gene regulatory program. That's the logic of that explanation. But in the lab, we had what I like to call a night science moment. Night science moment is kind of like a moment that we all like fantasize about. It's what we want in the lab, like a new idea. And our night science moment was actually that the cell can learn. That just like you have a brain, what if each cell has a little brain and it could figure out what to do in order to adapt to a new situation? What if it could do that? So in this case, there's a transcription factor, which is a kind of protein that is like a manager. It turns on other genes, called AP1. And it's been known for many years that AP1 is an important transcription factor, and there are tens of thousands of papers about this protein. But we're hypothesizing that there's a new function that still remains to be understood. And that is because AP1 senses stress and controls genes. And so why is that important? Well, AP1 has what is required for an evolutionary algorithm. So you know Darwin's theory of natural selection was transformative for biology. And it applies not just to the origin of species, but also when we study so many different processes. It has even been harnessed by biotechnology. And so we were very familiar with the theory of natural selection. What I want to propose is that that theory also applies to the cell, because there's a combinatorics of which kind of AP1 subunits occur, such that in a particular cell, there's like an ID, like a barcode, because of which dimers occur. And basically, what my model is that every cell has a particular AP1 pool, and that could change. In other words, that's variation, which is a crucial component in the evolutionary algorithm. And then there's feedback, because what the cell actually wants to do, in my opinion, is actually what we want to do in our life too, which is just to reduce stress. If a cell has a mechanism to always reduce stress, then it can survive. And so let me tell you this idea in a different way. I mentioned earlier that cells can learn. Cells can maybe switch to new states. So how would they do it? Imagine that a cell could change its AP combinations. There's a kind of combinatorics. And with each combinatorics, there is an effect of whether it reduces stress or increases stress. What I'm proposing is that with the introduction of each dimer here, each transcription factor, the cell can make a decision. Did it improve or did it worsen? It's kind of like when you play this game, hot and cold, with kids. You know, you hide something in the room, and then you say hot, getting hotter, no, cold, Antarctica, very cold. You know, and they find it in a really short path. So in our model, the cell is doing the same thing. It keeps changing things randomly, but keeping the changes that are reducing stress. So there's a certain feedback, and there's also memory for next generations. So what have I told you? I told you that, yes, you could think about each cell type as having a program, but don't be misled by this kind of Plato idealist framework. Really, cells are always changing states, and there's a lot of things to be explored for how cells can actually learn to adapt to new environments. In the little time that I have left, I just want to mention what night science is. So Francois Jacob, he earned the Nobel Prize with Monod for figuring out the principles of gene regulation. He said that, you know, what science is in the process, like when scientists are actually doing it, it's kind of like being an artist. There's what he called day science and what he called night science. Day science is executing. Like you have a hypothesis, you test it, you wear your white coat, you have controls. It's very orderly, predictive, logical, right? You have to be organized. So what's night science? Jacob says that night science, that's like the crazy part where ideas happen, the inspiration, a night science moment. Night science wanders blind, it hesitates, stumbles, recoils, sweats, it wakes with a start, doubting everything. It's forever trying to find itself, question itself, pull itself back together. Night science is a sort of workshop of the possible where what will become the building materials of science is worked out. So night science is that part where we still don't know what's happening. Like in the project that I explained, we still don't know how cells can adapt to something new. And I think the best way to explain what science is that you're always going in between day science and night science. Day science testing versus night science confusion and looking for a new idea. Oh, you have the new idea? Let's test it. And to a large extent, we scientists were always like pulled in between these two worlds. And I think that's the fun part. So my friend Martin Lurcher and I, we've been talking about this a lot. We have a podcast that you can check out where we talk to scientists and ask them, hey, Mr. Scientist, hey, Mrs. Scientist, how do you have ideas and what's your process? We have a manifesto, as we call it, that this needs to be taught, like that everybody needs to learn this. It's a crucial part. It helps scientists. It makes science more fun. And I think will attract more people to do this very important work. So thank you very much for listening, and I look forward to being in the conversation later on too.
Muchísimas gracias por esa gimnasia mental a la que nos ha sometido el profesor Yanai. A continuación,
And now I would like to introduce the next speaker, Dr. Vento. She is a leader researcher in the Singer Institute and the Cambridge Center Institute. Vento tries to understand how the cell microenvironment influences on the identity, the behavior, and the functions of the cell. Her lab combines spatial transcriptomics, sequencing of single cell, and the development of computational tools that allow us to interpret these communication cell to cell that is so complex. Her team has generated cell atlas of human tissues with a very special approach focusing on women. Dr. Vento has received the RC, the Welcome Lead, and the Sanger Initiative has been recognized with awards as Early Career Research Award and Michelson and Science Prize. It's a pleasure to receive now Dr. Vento. Hello, good afternoon. It's a pleasure for me to be here and introduce the work that we do in our lab. I'm going to do it in Spanish, but maybe I will say a few words in English as well. As I said, we have a strong interest in reproductive tissues, and we look at the reproductive tissues not only due to their function in fertility. Obviously, they are key for the progeny, but it's mainly for their endocrine capacity and how in that way they have the possibility and capability of communicating with all the tissues that we have in the human body, in the female body. In this manner, in the reproductive tissue do acquire a central role in the body of the woman. And in spite of this, we know very little about their composition, how they are developed, how they are regenerate every time during the menstrual cycle. And today I will tell you about how with these omic technologies of single cell we have made progress in that respect. In a more general idea in our lab, what we're interested in is answering a question that is very simple and complex at the same time. How the cells in the body, that most of them share the same genome, nevertheless have different identities. And in reality, how these different identities of the cells are important because they do the assembly with other cells, and they will form the tissues. And those tissues, that's where the function of the body takes place. And in reality, those tissues are, when we have pathology, that's where the pathology occurs as well. So in reality, in order to answer this question of how a genome gives rise to such a diversity, cell diversity, we use the technologies that I has been introducing before, the single cell genomics. And basically, as what Itai told us, it gives us information on the cell as a fundamental unit in the human body. Apart from those omic cells, we also have what is called space transcriptomics. Those technologies do allow us to measure the omics, particularly the transcriptomics, directly on the tissue in such a way that we can have the information of the cell we can link it with the information about the histology and also differences of how the cells communicate with the cell that it has close to it. And this is, as we were saying before, has given rise to consortia as the Human Cell Atlas, that is an international collaboration that wants to map all the cells in the human body. And particularly, one of the aspects that our group has been mostly interested in is in understanding the reproductive system. And when I started working on this reproductive study by chance, because I'm immunologist by training, what little we knew about those tissues compared with what we know in other tissues in the body. And when I started working on it 10 years ago, I realized how we yet didn't know the progenitors, how they were, the structural cells were differentiated from those who give the support. And this made that the molecular part and cellular part of the reproductive tissues was a sort of black box in our knowledge. In reality, this has a strong impact on health. More than 1/3 of women are affected by pathologies of the reproductive system. In the best of cases, this can affect your daily life and your quality of life. In the worst of cases, really, it has a strong effect on your life. And apart from all that, women are in a situation where the diagnosis for these diseases, tools for them, there are few, but it takes more than 10 years in the majority of diagnosing them, and there are not too many treatments that are efficient. Most of them are hormonal treatments, so they have many side effects, and not always are they efficient. And in reality, why is it so? Well, there are many factors involved. There has not been too much funding, and the science of the reproductive system in the human is different from what are the animal models as mouse models. But also, an important reasoning is that reproductive tissue is extremely dynamic, and extremely dynamic both in time and in space. The reproductive system is transformed by several organs, the ovaries, fallopian tubes, etc. Yes, thank you. And the vagina. And in reality, in the adult life, as we have those fluctuation hormones, this runs in cycles. The endometrium, the mucosa of the uterus. We know that every month is destroyed and then regenerated, and all those hormones that have an effect not only local but also at the systemic level. But it's a dynamic system. This dynamic system doesn't end in the adult life. It goes through the whole life of the woman, because those tissues are developed in the uterus but are not functional until puberty. They acquire many changes during pregnancy, and then they decline in their function during the menopause. And in fact, in each one of these periods, there are really hormones that are fluctuating. This is what we see, that the woman has to be considered as a complex covariate, not only as the opposite of being a man. And in reality, at this respect, all these technologies of single cell have helped to understand a great deal about all this complexity. And what happens? The human reproductive tissue is very dynamic, as I've said, and changes through different scales, from genomic regulation to the cell identity and the makeup of the tissues. And this goes through the rapid communication that happens, crosstalk between the cells. And
this respect, in order to better understand this whole process, both in the reproductive system and in others, our group has developed computational tools in order to understand how the cells have a crosstalk between them, obtaining those data. One of them is called cell phone DB, where in reality what we do is understanding the communication that takes place between molecules that are called ligands and receptors that are expressed in different cells, and that through them the cells talk to each other. But then also these other cases of reproductive tissue, the hormones have a lot to say. And what happens with the hormones is that many of them, as the steroid ones, are not proteins. So to infer, to make them is to reconstructing the whole pathway, the metabolic pathway that will then give rise to these hormones. And this is what we have done in another tool that is called single cell file. And with that, we can, and with the generated data, we can reconstruct this, rebuild this communication among adjacent cells and also between cells that are in different places in the body, as in the case of hormones. A little detail, more in detail, how we infer this communication cell to cell in this tool in cell phone DB. Basically, this tool, on the one side, consists in a database of those ligands and receptors that know each other and takes into consideration their protein structure, bearing in mind that in many of the cases you need more than one gene that has to be co-expressed so that this receptor is a functional one. Apart from that, there is also a computational method in order to ask or interrogate the data. And somehow there are several tools that we can use, including some statistical methods to see in what cells these receptors are expressed and those ligands as well, and also forms of filtering those interactions, for instance, seeing if if they coexist in the same space, or looking at the regulation of downstream regulation that goes inside the cell, where the receptor receives the signal, this becomes an active signal. Well, with all this, with all these computational tools, and with all those millions of data that have been generated, what we have done is generate those cellular atlases of the reproductive tissues across the lifespan of the woman. These cellular atlases are references like the genome is a reference for the DNA sequences. Those atlases are references for the cells. This means that when somebody in his lab produces, with this signal cell, produces new information of cells, they can be referred to that atlas to give it a background that of what we're seeing in the lab. But apart from generating those references that can be used by the whole community, what is even more important for us has been the amount of biological information that we have been able to extract from them. I will tell you some of the things that have been more fascinating for me. One of the things has been really to describe the progenitors and the forms through which those progenitors will give rise to all the cells that we have in the tissues. Particularly, one of the things that we have observed is that the immune cells play a key role in this function of the reproductive tissues. I will tell you more about it later. The other thing that we've seen is that the information is not only about genome, cell, and tissue. In the tissue, the cells are close to other cells, and they will communicate with them, and in turn they will inform about the cell identity. And what we've seen is that we have new cells that are part of that tissue that form signal gradients and that indicate to the cell where to go and what it has to become, what to become. And as I said before in these examples, we have discovered that the immune cells are structural parts of the reproductive tissues, and this includes we have discovered new macrophages that basically are a cell type, and in this case what they do is helping to regenerate after every menstrual cycle, help to the regeneration of the tissue repair, and guarantee that everything, all the vasculature is being formed and that the epithelium is also regenerated. And we have seen that in diseases as the endometriosis, those macrophages are deregulated, and we are observing that they do have a causal effect on the disease. Another thing that I also mentioned was that we have discovered new progenitors. One of the examples is in the gonads, and one interesting case is that at the beginning of our lives, we have progenitors that can be differentiated both in the testicles as in ovaries. And we have discovered that progenitor that is the one that will decide if the gonad altogether will be formed as a testis or as an ovary. And we have also discovered progenitors of where the follicles are formed, that where the ovarian reserve will be. And this is important because in the humans we are born with a given amount of ovocytes that will not proliferate later on. This is another example of other progenitors, in this case, in the case of the endometrium. All this layer regenerates every month, and these are the progenitors that we have seen. And we have identified that every month there's a renewal of the whole tissue. And as I said before, all the different gradients that make compartments in the tissue in several subunits, functional units, that can't simply be appreciated by the morphology, but that are really very important because they are going to determine what the cell is going to be. And during the development in the reproductive tissue, at the beginning we have a tissue that is very homogeneous. Little by little will be differentiating, giving rise to the different tissues. And we have discovered all these gradients and how they are formed through the development in order to give rise to the different tissues that will have a different function. And all these gradients, we can also see them in the adult tissue in order to be in the same background as the endometrium. You can see all these different gradients, and all these are signals that are going to drive how the tissue will be differentiated, and it differentiates in a very organized fashion. And finally, one of the important advances has been to use all this information of how we understand the tissue in order then to go to the lab and make tissues in the lab that we can make them grow and use them to then validate, for instance, make the alterations that obviously we can't do on the humans. This is important for the reproductive tissues because, as I said, the mice really don't characterize the whole physiology that takes place in humans. The idea is to use all these models, these atlas, as a recipe in order then to recreate step by step this organ in the lab. And with this, we have been able to improve the models that we have in the lab today in order to study the reproductive systems, but also making new protocols in order, for instance, to generate B cells directly in the lab because these are the cells that generate the antibodies. And with all this, and I come to the end, what we have been doing through all these years has been create a sort of framework that allows us to go from what we see in the sequence to the cells and to the tissues. And implementing this to health for women, what we do is making biology of women into a science that really can quantify, that we can model, and that we can predict. And eventually even we will be able to manipulate. And with all this, I would like just to thank all the members in my lab that have been working together in order to make all this possible. All this science, and it's very fun to work with them. Thank you very much.
Muchísimas gracias.
Thank you very much, Dr. Vento Tormo, for that fantastic talk. Impressive. Very necessary, all these studies that you have long overdue. I would like to introduce a new star of science here in Spain that will talk about a different aspect. Dr. Maria Colometache is a researcher in the Ludwig Maximilian University in Munich and German Environmental Institute. Dr. Colometache is specialized in epigenomics and the study of cell heterogeneity. Her research starts from a fundamental idea. Although all our cells share the same DNA sequence, their identities are surprisingly diverse. And the work in her lab is focusing on how the different epigenomes are formed, how the epigenetic changes are, and how stable they are, and how the epigenetic changes are translated into different morphotypes and phenotypes from the cells, including the pathological ones. Her team develops new computational methods and mathematical models able to study and detect how the epigenome changes in specific situations and how these variations do alter the cell function. Thank you to the combined use of combined genomic transcriptomic data of individual cells. Her group has now become the reference point in the quantification of heterogeneity within cell populations. So it's a pleasure for me to introduce Dr. Colometache. Hello, thank you very much for this introduction. It's a pleasure and an honor to be here with you. It's more than 20 years I left from Spain. I will try to give my talk in Spanish. Let's see what happens. The important thing is to have the pointer. So, as Erika said, in my group, we dedicate to develop algorithms. I'm a physicist for training. I went over to biology a few years ago, so I'm going to tell you more than anything else the methods that we have developed and how we can use those methods in order to analyze then the single-cell data with a special emphasis on the single-cell epigenomics. First, in order to motivate the audience, I would like to say, what is the epigenome? Why are we interested in it? With a very easy, simple example, very close to all of us. Here we have a cell, a single cell, and this cell is a zygote. This cell will differentiate and will generate a multicellular organism with organism with billions of cells and many different cell types as well. So what we know is that the zygote, as each one of the cells in this organism have exactly the same genome. This means that all these cells have exactly the same genes. But those cells are very different among them. They have absolutely different functions. We have the blood, the heart, the brain, many, many different cells. The question is, how come it's possible that from one genome that has exactly the same genes, we can generate a whole type of different cell programs that have different functions. And the answer to this is the epigenome. What we know is that we have the same genome but different functions because the DNA is not a molecule that is alone within the cell. The DNA is interacting with many proteins, and those proteins and the DNA in itself have chemical modifications, and this makes that different parts of the genome can be read and transwritten different manner, differently transcribed. That's why this part here is very open. Here we have a part of the genome that is open. So then the genes that are in that part of the genome, we can read them, we can transcribe them, and we can also form proteins. This is what we call open chromatin. There are other places in the genome that are absolutely condensed. Something that is condensed, it's impossible to access that part of the genome, so we can't produce protein. And what we also have is the small blue dots. These are chemical modifications in the genome, in the DNA. So those chemical modifications do also change as how we read the DNA. We know that when there are many of those chemical modifications, it's very hard to transcribe the genes and generate a protein. And that gives us the response. Due to all these different chemical modifications that we call the epigenome, we are able to produce many different functions from the same genome. And this is how the definition of the epigenome, all the different layers of regulation that allow us to generate many different transcription factors through the same genome. Thanks to the epigenome, we can generate many different cells. Of course, if we have problems in the epigenome, this is going to give rise to a disease. Only problems in the epigenome are often the origin of many cancers. And thanks to the measures of epigenomics of single cell, we can start understanding the heterogeneity that exists in many systems and many organs. For instance, a very complex organ, if we measure it as organ, we only see the median of how the epigenome is in the organ. But in this organ, we have many different cells. If we measure the epigenome, we can see what each one of these cells is doing. An example that is very much used is the one of cancer. When we have a tumor is not that all the cells of the tumor are exactly equal. If we don't measure it at the level of a single cell, we can't see how many different cell types we have in a tumor. And when we do give a treatment, we can't really reach one of these tumor cells. So then, from a cell, we can measure today at the level of single cell many different layers, out of which we have been the one we have been talking in the previous talks is the last one, that is the transcriptome, is the expression of the genes. We have the genome at the beginning on the opposite side, and then in between we have those layers that are the methylome, and if the genome is open or closed, that is chromatin, and also I'm going to call ataxy is the technical word. And what we do in our group is develop methods. Then through seeing those layers of the methylome and the opening of the genome, we can correlate, like with the same genome, we can predict so many different transcriptomes. So how can we use those data of a single epigenomic cell to study the different cell types? How do we do that? The first question is, what part of the genome are we interested in? In all the talks that we heard until now, we talked about the transcriptome. So now when we talk of the transcriptome, we have the genes, and we quantify the expression of each one of the genes, and that's it. We don't think further about the rest of the genome. When we measure the epigenome, we are measuring every cytosine, adenine, guanine, etc., that are in the DNA, and we can't manage it from the computational point of view. So we have to think, what is the part of the genome that we are interested in? The genome is made up of many different units. We have the genes that are going to be transcribed. We have the promoters in front of the genes that are important for the transcription. We have the enhancers that will increase the expression of the genes, etc., etc. So we have to decide how we are going to quantify all this. In general, when we analyze epigenomic data, what we do is either use this annotation of the genome, or we are going to use fragments, segments of the genome. We say in these 1,000 bases, there is a lot opening, or the genome is very close, and in the second, etc. So that's how we quantify, and then we take the data of the single cell, and for each one of these units, we measure for each cell how much opening there is, or how much methylation of the DNA we have. And thus we build at the mathematical level, that's how we work in my group, we build matrices that are going to have the cells, and in each one of these units, the epigenome as well. So we have already made a complex problem in something that is much easier, many mathematical matrices. What can we do then? As we've seen before, what we do is measure how similar are two cells among them, as we do with the transcriptome. But in this case, with the epigenome, we obtain a distance between each pair of cells, and then we do exactly the same that we've seen in the first talk. We go from one space that is multidimensional, 20,000, 500,000 dimensions, to a space that we can understand as humans that has, for instance, 2 dimensions. So each dot in this plot is a single cell and is surrounded of the cells that are most similar among them. But in this case, not from the transcriptome, but from the epigenome. So I've said already that the fun with epigenome is that we can look at the same cell, but in its enhancers or in the promoters of the genes or in the genes. So what we can do in this case is that we can basically look at the same cells. In this case, in this example, it's the brain of a mouse. These are neurons. And we can look at the same cells, the same neurons, from different points of view. So each dot is a cell, and the cells among all these panels are exactly the same. But here I'm looking at it. I'm looking at windows in the genome. Here I'm looking at the amplifiers of the gene expression, and here I'm seeing the promoters of the genes, and here I'm looking at the genes. And certainly, a simple side, you can see what is the best representation to describe those cells. In this case, will be the amplifiers of the expression. But we can enhance it. But we can also start comparing the cells that are this group, this orange group, for instance. What do they correspond to in the promoters? We can start seeing if the cell types join each other according to what part of the genome we're looking at. And this can be done with the epigenome, but not with the transcriptome, because we have one transcriptome. So we can start by comparing them among them. And then the last question is, but what cells? What cells are we talking about? What do we do in order to make cell type identification? We look at what part of the epigenome are the most different among these different groups that I have put here with different colors. So what we do are some statistical tests in which we look at the whole epigenome. We compare it between different groups, and we say, look, this position of the epigenome is very different. It's very open here and closed here, for instance. And what we are going to do is a sort of ranking of the positions of the epigenome that are more different among the groups. And in the example I'm going to give now, I'm going to do that with the promoters of the genes. In reality, in this population, it's more interesting to have, it would have been more interesting to do it with the enhancers, because it was the one that best defined the cell type. But we are still discovering a lot of science. The enhancer is very difficult to know exactly what gene they amplify and what they are exactly doing. The promoters are much easier, much more linear. So what we do in the lab, for instance, is to take those cells. We know that they are all neurons. We divide them, in this case, only in 2 groups to facilitate the problem. And we ask what are the most different positions between both groups. And we are not biologists. We talk then with the biologist and we tell him, I have this, can you help me to understand what it is? And they say, oh, here you have neuro2 gene. This is very important. We look at it better. Oh, yes, look at it. In the blue cells, the methylation is low. In the orange cells, the methylation is high. There is a clear difference between the 2 populations. and they tell us, yes, but this is important for the inhibitor cells, etc. Okay, then the orange are going to be the inhibitor ones, and the blue, the exciting ones, excitatory ones. This is more at the level of classical statistics, but then we also have more modern methods based on machine learning that can also allow us to access other parts that maybe are more difficult to access using these the classical statistics. Something that we've done recently is look again at one of these multi-dimensional matrices, the cells, the positions of the genome. And here what we have done is putting another variable on top. We have some vectors that are very long, 500,000 of dots. And in principle, those vectors could occupy any point in space. But what we know is that if a gene goes up, the other one is going to go down. And if a gene goes down, the other one is going to go up. So then we know that those data don't occupy any possible point in the multidimensional space. They occupy a sort of plane. And if this goes up, this is going to go down. So we have a biological constraint that makes that our data are in manifold. We learn how the geometry is of those data, and when we go to this low dimensional, the small points in 2 dimensions that I showed to you, what we ask them to preserve the geometric structure that this space, multidimensional space, had. This is as if it has more power, more capacity to detect changes. Then we apply a classifier, and what we do here, explainable AI, that is open the black box that intelligence and artificial has, and ask the model what points of the genome are the most important to classify a cell in one way or another. And from there on, basically, we do learn new cell markers. With the statistical classical methods, it would have been impossible to detect them. At the level of machine learning, we have many new methods and new ideas that can be implemented with these data. But basically what we do is that we have the dots for each one of these groups. We have a list of different positions in the genome that are important. We talk with the biologists, and the biologist tells us, aha, this is that cell type, and this is that cell type, and we write it down. Basically what we do is with epigenomic measures of single cell, we can also have this low dimensional representation, and at the end we can see what are the cell types, and here learn a little bit more as to the regulation of the genome and why different epigenomes give us different cell types.
In the last part of my presentation, I'll explain how we can use epigenomics to study the heterogeneity of the genome of single cells. This is highly relevant in cancer. You know that each cell bears 2 copies of each chromosome, but in cancer, the majority do not have 2 copies, but they have an absolute chaotic mess. Too many chromosomes, too little, and the majority of tumor cells have this kind of problems with a huge heterogeneity, sometimes aneuploidy, sometimes duplicity of the genome. So if we study them at the level of single cells, we will be able to understand why with treatment some cells respond and some others do not respond to treatment. So what is it that we do? We map the information available for every single cell in the genomes. If there is a gain of a chromosome bit, we gain more data. There is an additional quantity of chromosome, and then we use statistical methods to elucidate the difference. Here, a change has occurred. This is just a cartoon, a diagram, right? And these are the real data, obviously much more complicated. And we develop computational methods for analysis. In this case, we apply the binary segmentation, which is a very typical methodology to see changes in a signal in order to identify the breakpoints in a genome sequence. Where are the changes taking place in the genome signaling? There are many methods, but we look at different positions. We compare left to right, and then we determine the distance in between the distributions. And when we face the breakpoint that we identify, this is the largest distance. So we identify the place where the change takes place, and then we see whether it is a gain, a loss, or an ordinary amount of chromosomal material. This is what we do for a single cell. And then with the tumor, we have 3,000 cells. Each line represents a cell. In green, we have normal openness, the mean openness. In red, the gain. In loss, in purple, bluish. And as you can see, there's many different cells and different profiles. Here's a small example. This is from other laboratory, but you can see this is a basal cell carcinoma. Sorry, I don't know how to say this in Spanish. It's about 2,000 cells, and we have the epigenome of each of these 2,040 cells. First, we analyze the cells to determine its cell type. This is the representation, and in blue we see, okay, this is the cancer. And in orange, these are the associated fibroblasts adjacent to the cancer. And this is the kind of basic function of our cell type. And then we ask them, what about genome analysis, chromosomes? Are there losses, mean openness, or gain? Well, for each of these cells, there is a different profile. Some parts have lost, some parts have gained. So we can group them together by similarities, and then we ask ourselves, of the tumor cells and of the fibroblast cells, which correspond? And we can see that fibroblasts in orange are basically two types. And yet the tumor is absolutely heterogeneous. Within the tumor, we have a myriad of different landscapes when corresponding to losses or gains of bits and pieces of chromosomes. But if we do it the other way around, and if we query out of the different profiles, where can we locate them? When I look in the low-dimensional embedding, well, I see that they are completely mixed in between. So if I do a classical analysis of my tumoral data, I will not be able to determine that the genome composition is very different for each cell, which is the reason why the cells respond differently to the therapy. So with this methodology, we add an additional layer, an additional information to the experiment, because otherwise it would just tell you what they are doing. But now we can tell what the genome of the cells is about. So I hope that you can understand that epigenomic data is a very important regulatory layer. Between genomics and transcriptomics, we can generate multiple transcriptomics. Starting for a single genome is the transcriptomic layer, and that we can also use it to understand better the genome of the single cells that we start with. Here are my groups, my thesis and postdoc students, and thank you very much for listening.
Muchísimas gracias a María.
Thank you very much, Maria. That was a very stimulating presentation. And now, before we start the debate, we get to the last speakers. I'm sure you're going to have many questions, and there will be time during the debate and some Q&A for your participation, of course. And to conclude today's session, it is a great pleasure to introduce Professor Holger Hine. Professor Hine works at the genomic laboratory at Barcelona. He's an international reference in single-cell genomics and spatial sequencing, and he works with biology and computational biology to develop tools to be applied to basic and translational research. He's a member of Human Cell Atlas with profiling of the immune-mediated cells and the development of new strategies in immuno-oncology, immune diagnosis, inflammatory diseases, and clinical applications of the single-cell genomic techniques, and he is a member of the ICRIA. as a co-founder and medical director of Omniscope, he fosters the clinical implementation of cell profiling of high-resolution techniques in order to improve upon the diagnostic and treatments that are immune system-based. The immune system is a cross-sectional modulator of disease, and that's what he's going to talk about in his presentation. Thank you. Professor Hine, you have the floor.
Buenas tardes a todas y todos.
Good afternoon.
Lo puedo intentar, pero mejor que no para todos. So thanks for the very kind introduction. It's an honor to be here, especially after this amazing lineup of speakers, I have to say. Today I will introduce you to the power of a million cells, or even millions of cells. And as Itai already mentioned, the early days of single-cell research were very different from where we are today. The early days were really using individual cells, positioning them into a plastic plate, 96 at the same time. So we were able to analyze hundreds of cells. And today we are able to analyze even millions of cells from a single sample. So the scale is really changing, and that means it allows us to do more things, to look at complexity, and even complexity over time. And I will talk about those projects. And I will talk about projects that allow us to profile millions of cells, focusing a bit more on the technology. And I will talk about atlas generation. So we are here in the Human Cell Atlas meeting, and we've been contributing over the last years to generate an atlas of the healthy immune system, but more recently we published the atlas of the inflamed immune system, so integrating data sets not only from healthy individuals, but from many patients suffering from immune-mediated and inflammatory diseases. And I will conclude looking at my own immune cells that I've been profiling over the last 5 years with technology developed in a company that I founded 5 years ago, Omniscope, where we now use the immune system as a biomarker and immune cells and circulation really as living biomarkers with technology that now really allowed us to profile, in my case, more than almost 70 million cells over the last 5 years. But I will kick it off talking a bit more about technology, because profiling not 100 cells, but millions of cells can be very, very costly. And in our lab, we were working on technology that makes the scale much more cost efficient. And we developed a technology called single-cell transcriptomics analysis and multimodal profiling through imaging. And you already realize that there's no sequencing in this equation, because this is exactly what we wanted to remove while we profile individual cells, to make the entire process, profiling millions of cells, much more cost efficient. Okay, there's no pointer, right? Okay, so this is the classical way single-cell sequencing has been done over the last decade, moving from plates to droplets. So these are technologies how the Human Cell Atlas and most of the projects developed the data today. And in the end, we have kind of two individual processes combined. So we have the microfluidic part, that is encapsulating single cells in individual droplets. And at the end of the process, we put those droplets together and we generate sequencing-ready library. And at the end, we do sequencing. And on this side and on this side, we have proprietary technologies. We have very costly reagent. So the entire process is not scalable with a given budget in hand. So what we thought of doing is taking sequencing and microfluidics out of the equation. So in the end, what we do is replace sequencing by imaging. And José already introduced spatial transcriptomics as a method, and we do basically a life hack here, not using spatial transcriptomic methods to profile tissue sections or tumor sections, but we actually bring cells in suspension, for example, blood cells, onto those glass slides, and then we use an imaging readout to do single-cell profiling. How that technically looks like, in our case, we bring a lot of immune cells onto those glass slides. So we have a very cheap glass slide in the start of the process. We can mount millions of cells at the same time across these glass slides. But we can also generate sub-areas, so-called substamps, where we then can multiplex many samples from many donors, patients, for example, still profiling thousands of cells at the same time. And at the end, we are not sequencing, but we use commercialized spatial transcriptomic devices that in the end would create an image of those glass slides and cells on those slides, and would take an approach that is probe-based imaging or probe-based gene expression profile, which is in the end much more cost efficient than the sequencing-based readout. And in practice, it looks like this. So you have a glass slide, you have a monolayer. In this case, in our case, immune cells isolated from the blood, so-called peripheral blood mononuclear cells. You have T cells, B cells, monocytes, dendritic cells. So everything is in there, but presented as a monolayer. And then as the next step, we can use these imaging-based methods, which today scale to full transcriptome analysis. So we can read 18,000 genes using an imaging readout, and we can do classical imaging-based analysis. On the top, you see an H&E stain, so we can look at the morphology of those cells. And then we can combine it with the imaging readout, which first of all is segmenting the cells. So we have markers that define the boundaries of the cells, so we know where they're located. And then in the next step, you have the full transcriptome readout. And with this, we can create a full map of immune cells that are on this glass slide, and you're basically digitalizing. So you put a transcriptome analysis, and you can do cluster analysis. You can do the very classical readouts as we've done before, using single-cell genomics tools and tools Rosaire and Maria described. But at the beginning of the process is no sequencing, so it's very cost-efficient, being done through imaging. And this is just one example. So these are the so-called UMaps, two-dimensional representation of immune cells, now 1 million read from a single slide. And we can group them together. We can use that transcriptome profile to generate atlases. What we were doing here, we were kind of, just to give you an idea of the sensitivity and specificity of the methods, we were hiding single cancer cells among those immune cells. So all of these are immune cells, and there are very few cancer cells hidden in between. And just looking at the morphology here, you would recognize, okay, this cell is much bigger than the surrounding cell. But this cell, as we now have the transcriptome information, we could say this is also expressing EpCAM. So EpCAM is a biomarker for cancer. The surrounding cells are immune cells, they are not expressing this biomarker. And then, first of all, we have generated transcriptome information, so we can take the entire transcriptome of that cancer cell and we can profile and sensitively and systematically detect those in a given sample. So this is exactly what we've done here. And from the diagnostic part, what we're simulating is really the analysis of circulating tumor cells. So we have a lot of immune cells, we have very few cancer cells. In this case, we have 30 tumor cells hidden amongst 2 million PBMCs. And we can look then at the morphology. So we can identify and combine morphology information. You can combine it with transcriptome information. And basically what you're doing is kind of looking at looking for the needle in the haystack by combining this information, combining information that then lead to the identification of those very few cells. And this is, in our case, a simulation of circulating tumor cells and the identification using STEM. This is something that we are pushing now towards clinical application. As already mentioned, there's no need to put 3 million cells on the glass slide. You can generate substems, and in our hands, we generate up to 96 substems per glass slide, and with this you can multiplex a lot of samples, you can multiplex donors. In this case, as a proof of concept, we were multiplexing cancer cell lines, and then again, for each of these substems, you will have thousands of cells, so good representation about the heterogeneity. And then at the end, we can do classical single-cell analysis looking at transcriptome profiles and states of these cancer cells over time. So this was kind of defining the technical aspect and looking a bit into the future, how to generate even larger atlases more cost efficiently. One second. In the second part, I would like to switch gears to atlases we've been generating in the past, mainly through our contribution to the immune cell atlas. So immune cell atlas is really not only taking cells from the blood, but taking cells from different organs, from different tissue sites, and generating a kind of comprehensive map of immune cells in the healthy human system, which is the mission of the Human Cell Atlas today, generating atlases, blueprints, reference maps of the healthy system. But we've gone a step further already, not only analyzing a reference map of healthy humans, but we were actually combining those with different inflammatory conditions. So at the end of the process, and talking about millions of cells, we had about 6.5 million cells combined across more than 1,000 donors. And these donors were not only healthy individuals, which defined our reference, but patients suffering from different inflammatory diseases, mostly autoimmune diseases, but we added some disease indications that would also stimulate the immune system, for example, chronic or acute infections. We added some cancer cells with the idea really to map out the full plasticity states of immune cells in the human system. So by profiling then more than 6 million cells, we were able to say, okay, this is more or less a comprehensive representation how immune cells can behave in humans in health and disease. Because we mapped out basically activation states in very different environments challenging the immune system. And you've seen these UMaps before. Now they're getting more and more messy because we're integrating more and more cells. But in the end, what you can do then is do this clustering that Itai already mentioned, looking at cell types and cell states. So high-level types like B cells, T cells, monocytes. You can group them at higher level towards the activity states. But having so many cells, you can actually go much deeper to a next level where you look at activity states that are very, very detailed, very fine-grained, and some of them already related and specific to their underlying diseases. So there are diseases that push those immune states into specific activation profiles, and then are also indicative or biomarkers for the diseases themselves. And this is actually the path we were following here. So we are then generating a map of immune cells and immune activation cells. And on top, you see the different, let's say, inflammatory profiles or processes of immune cells. Then in the rows, you see the different diseases. And here at the bottom, you see the different immune cell types, high level. And what we're looking for is really at patterns. So for example, diseases or groups of diseases where in this case, these are CD4 positive naive cells. So we see that those are activated in their chemokine receptor process, not only one disease, but in many diseases, and most of them being inflammatory diseases. We can look at the horizontal patterns where we have, in this case, a single disease, one process, in this case, interferon induction, represented across many cell types. So it's not one single cell type driving diseases, but there are many contributing, though with the same immune activation program. And then we look at patterns where we have a vast representation, for example, almost all immune-mediated inflammatory diseases across monocytes and dendritic cells were activated in this process related to TNF-alpha. But in the end, what we're describing here are potentially driving mechanisms of the diseases, but we are also kind of looking at fingerprints of diseases in the blood, because it's not clear, or it wasn't clear before, that when we profile blood, it really is a representation of those diseases, and that blood cells themselves have enough signal, so enough transcriptome profile change to really represent those diseases. And this is what we're asking in the next step. So don't look at this slide at all. Just have in mind we're generating the maps, three-dimensional representations, embeddings of the full representation of immune cell plasticity. And then what we're here aiming at is really using immune cells as living biomarkers. So we take a blood sample from a different patient that were not in the training set. We map it on top of the atlas, and we are guessing, predicting, classifying what this patient is suffering from, which is in the end the goal of universal diagnostics using blood, that you predict from a sample what that patient is suffering from. And having an atlas trained on very different diseases, that's kind of the ultimate goal we're getting here. And what we've done in the process is basically generating the maps, mapping external sample on top, and we were challenging ourselves with 3 different scenarios, being one more challenging than the other. For example, leaving samples out from our training set, leaving cells out, or leaving entire cohorts out. And what we're then simulating is a different kind of complexity, how to develop such diagnostic tool and how to implement it in the clinic. And what we're finding here, so what you have on the x-axis is basically the accuracy we are predicting from an outside sample, from a new sample, a test sample, the disease the patient is suffering from. And the scale goes from 0 to 1, so 0.9 something is very high. So we're very good in predicting the origin, so the disease of the patient. And this is working almost perfectly in the scenario 1, which is called 5-fold cross-validation. So we are kind of using our training set and testing it within the same set of samples. In scenario 2, we were leaving out patients from the training set, but still they were present in the same cohort of patients we were profiling. And also here we generated very high accuracy, and what you can see, what I didn't mention here, that you find these fingerprints of diseases not only in very specific cell types, but actually across cell types you find the signal, meaning the disease itself is leaving fingerprints not only in T cells or B cells, but somehow in all of them together. And if you take a majority voting system, you're actually very good and precise in predicting. So this was very good news when we left out samples and we predicted them in, but the method or the approach completely failed when we looked at fully external samples. So samples the training set has never seen, and samples or cohorts, let's say centers, institutes, data that the training set has never seen, the method completely failed. Meaning in our training, in our representation, we're not fully representing, or we might fully represent the complexity of the immune cells, but we also represent something else we call batch effect. So something is a technical artifact introduced by not being perfect in balancing samples, having many of one disease from one center, profiled with one technology, not the other. So there's not a perfect representation of technologies or technical artifacts. And that's really is bias in our prediction, and it makes us fail in these very challenging, let's say, diagnostic clinical prototyping. And this is what we're following up now, building an even larger consortium with many European genome centers generating data at different sites repeatedly. And then we have clinical data or clinical samples from around 20 different sites generated repeatedly by the genome centers and AI experts then to generate an even more robust model of the immune system. So this, what I just described, is basically our work, how we would kind of connect and plug into the clinical side as a clinical translational effort. But I would like to wrap up in the last 3 minutes or so, really looking at a more personalized view. So what can single-cell transcriptomics, single-cell genomics do for you and me today? So what we have been doing and developing with Omniscope is really a set of tools that allows us to use technologies such as single-cell analysis to generate a very comprehensive representation of immune cells. And we're very much interested in the aging process because when we get older, the immune system stops functioning. We lose kind of the immune control. We're less protected. Immune cells get inflamed. They cause chronic inflammation and tissue damage. So this is also something you want to measure because it's something you want to control for. So when I exercise more, I know my inflammation should go down because something that is very much proven, but I need to measure that. And for that, we've kind of built a platform called Lifetime that does measure features like inflammation, immune age, resilience, meaning protection to common viruses, through the specificity. We look at senescence, we look at telomere length. So really all these hallmarks or biomarkers of aging that are changing while we age, and that what we want to kind of tune down or avoid in the process. And another thing that is important is then to look at this in not only as a snapshot, as a baseline, but in the context of time. So how does it change while I get 2.5 years older? In this case, does my predicted biological age change? And when I do intervention or when I get sick, how does it accelerate or decelerate that process? And this is my own profile. So I was back then like 46 years old as a chronological age. I've decelerated immune age in general. That's very good news. And following that over time, you see there's some variation. And at this very specific point, and now switching to a more scientific view, I was suffering from Lyme's disease, so tick bite infection bacterial infection that caused actually a deceleration of the immune system. So you see a drop of my immune age, which is a bit counterintuitive because I was suffering from a disease. But in the end, an infection is nothing as in a training of the immune system, such as a vaccine. So that's something important to think about. Maybe vaccination is something positive, not only protecting us, but also has a more kind of secondary positive effect on the immune system. But then having single cells profiles and their transcriptomes, we can look at processes that correlate, for example, CD4 T cell activation, while suffering from the disease and going back to baseline after treatment, but also age-related process, for example, senescence, so-called zombie cells, where cells, they lose their function, still they're not dying, and they cause chronic inflammation in the process. So all this together is now helping me a bit to monitor my own immune health, but also to really look at interventions such as change in exercise, diet, or even supplements. And with this, I hope I could convince you that millions of cells are the solution for everything. And I would like to thank my wonderful group at the SENAC and the team at Omniscope, and thank you for your attention.
Okay, muchas gracias. Ahora quisiera...
Thank you very much. And now I would like to call on all the speakers, please join me with the headsets for the translation.
Us, with these four brilliant scientists, reflect on some of the great findings we've had in the sessions, and I encourage you to think about your own questions. So I will get started with a general question for the panel, but I would really appreciate if you want to ask questions, and you can ask in English or Spanish. The speakers have a translation device, so they'll be able to understand your questions. So that was fascinating, and in my view, one of the key things we've really heard from all your talks in their diversity and richness is that there is a huge continuum of diversity in cells, right? We heard about adaptive states and how cells change continuously. We heard and saw the heterogeneity within a cancer. We've seen now, the richness of our immune system or the endometrium. So there's clearly much more diversity than we originally thought when we thought of a cell type. So my question to all of you, maybe you can all sort of say a few words about what you think, is whether we need to continue thinking about cell types, whether it's a positive tool for biology to classify things in that way, or whether it's hindering our capacity to think about perhaps treatments, diseases, or evolution. How do you see it from your own lens, in your own work? Do we look to reclassify cells, right, with a much more multi-omics view now and agree on a consensus of cell A, B, C, or do we kind of want to loosen that definition because it's going to help us advance biology and medicine faster? So yeah, I'll pass it on to the panel, and then there'll be an opportunity for your questions later.
So in my view, I wanted to be heard. So in my view, this is just a terminology, so I think it depends on your biological question. So I think there will be some question. Obviously, I think the biology is complex, but at the same time, for some specific biological questions, we need to simplify things, and our brain is used to classify things. So it's easier to classify things. For some others, it's better to consider the reality, which is more a continuum. So I don't care much about how I think as long as we know that this is a continuum, if for some things helps doing some cell type classification and doing in a hierarchical way, so you will always be able to go to the step further, depending on, I think it's okay to classify. So yeah, it really depends on what your question requires. But certainly we need to start thinking about things as a continuum as we develop methods and all of this. But yeah, I am quite flexible. I am not that strict about classification, if that makes sense. I don't need to pass the mic. I was like...
As I'm speaking already. No, I think that the valid of cell types is still kind of important. Especially in the end, we generate atlases that are catalogs of cells, and then we need to talk a common language how we name things. And therefore, we need some kind of hierarchy, some order to that. And obviously, in the way the immune system, the human system's developing is that you have very discrete states that are not transdifferentiating. But then looking at the types, the cell types separately, we have to kind of appreciate that there's a lot of plasticity, there's development, there's differentiation, there's lineage commitment, and this is what we have, or what we're mapping out. Before I was asked the question, are we there already having mapped 300 million cells? Do we have the full overview of cellular complexity in our human cell atlas that defines our human system? I would argue that most of the major building blocks are defined now. They're probably very rare populations that we didn't catalog yet. But it's where we're now able to position these maps into these buckets and into these catalogs, really to use that with the community to talk a common language. But then also when we look at diseases to see, okay, where is the driving mechanism? So what is a cell that is responsible and driving disease themselves?
Yeah, just to follow up, I think it's kind of like this age-old question, to what degree does nature subscribe to our classifications? You know, we humans, we like to classify things, and then the question is like, does biology agree? And I think, you know, in a sense, the way I think about it is that we're drowning in data, but we're still very thirsty for principles to understand what we're seeing. My kind of bias is that, for example, if you talk about cell types, that there is a reality to it. It's not a figment of our imagination. Like, there really are, this clustering really does happen. And I think we need to focus more, at least what I tell myself is, I like to figure out the principles, like why is that? Like, what deep constraint is there biologically? Is it in the chromatin? Maybe it's methylation states, maybe it's some kind of transcription factor regulation, but this clusteringness that we see, it has degrees of plasticity, but I think we're still far away from like a general theory, and it would be great if you know, young people join our efforts, and then we get a general theory that explains to us why we see this reality of a cell type, but also the variation.
So I'm not going to add much, Leo, but I think that cell types are useful because we need to talk to each other, we need to give names to things. I think the deeper we dig, the more refined we measure, the more cells we have, the more, whatever we call it, cell subtypes, cell states, the more we discover. And I think it's normal. The more we measure, the more precision we have, the more we see the differences that before we were missing. And I think that maybe coming from a very quantitative background, what I would want to see is that we could quantify a little bit all of that. Like, maybe the people that work with immune system immediately call cell subtypes something that people working in brain would still call cell type. Can we at least somehow try to quantify some distance starting from which we would say, no, these are two different cell types, no matter whether I'm talking to an immunologist or I'm talking to a pulmonologist? Or can we try to study trajectories that would tell me, no, these are fully differentiated cells that would never transition to each other. They are completely disconnected, while some other cell types or states, or they are connected because they can transition to each other. I think if we could quantify all of that, which is super complicated, would also help us to put some order into what we now, basically now we use names, type, subtype, state, that do not really mean much. So maybe then we could at least put a meaning to these terms, I think.
So I'd like to give an opportunity to the audience to ask some questions. There'll be some microphones coming in.
Congratulations. Fascinating all the things you are working on. And I have a question because I was listening to Priscilla Chan talking about the Chan Zuckerberg Foundation, and she said that maybe by around 2030 we should have the human cell atlas more or less finished. And she even mentioned working on a human organ atlas, which I think is even more interesting, going from cells to organs. So what is your take on that? Because maybe you could get some funding by the Chan Zuckerberg initiative.
Yeah, I can take that, because I would, or the HCA Human Cell Atlas community would, or is arguing that we actually finished the first phase of the Human Cell Atlas this year. So we're celebrating the 10th year anniversary, and there's a meeting in June where all of the first versions of single-cell resolved atlases are being published. At the same time, we're initiating a next phase, the Human Cell Atlas 2.0, where we are moving technology that we've been using in the past, single-cell resolved, to something that is spatial resolved. So we're now focusing on spatial transcriptomics methods that help us mapping out not only the complexity of human tissue, but also the architecture and organization. And I think it's very much in line, what you just mentioned is that the moment we have a spatial view, we're technically mapping the spatial representation of cells in tissues and organs. At the same time, we're predicting the function of organs, and in the end, we're puzzling that together, creating a spatially resolved map, organ-resolved map of the healthy human. I think that can go further, but basically the first phase is being completed now, having a representation, and now we're putting things in order in a two-dimensional space.
Anyone else? Yeah.
Maybe I can add one thing. You know, there's, I would say, like 2 different ways to do science. And that is one, like an atlas, international collaboration, multi-teams, coordinating. You can achieve big things like the Human Genome Project. You could put people in space. It all requires large-scale coordinated efforts. But I think that, you know, there's something that I hope we don't lose, which is the power of individual labs working in a non-coordinated way. You know, if we look at the past discoveries, like where did CRISPR come from? That was a great discovery. Look at
From Spain. I'm from Spain.
That's right, Francisco Mojica. That's right, in the south of Spain. Yes, he was robbed of a Nobel Prize, and I'm with you on this one. But yeah, let's take the case of Mojica. So this was not a coordinated effort. This was an individual scientist working with their team, and there's a lot of breakthroughs that happen. And I just hope, in you know, with all the thrills of a large project, we don't lose sight of funding for the very important individual, maybe sometimes chaotic and disorganized, but ultimately resulting in very fruitful research.
Any other question from the audience? Maybe.
Hi, very nice talks. I have a more philosophical / biological question. There is something I have been thinking since a while, since my PhD, which is, for example, when people talk about different molecular dynamics like transcription, replication, and all chromatin changes, a lot of people say, for example, in terms of transcription, there is a lot of transcriptional noise, meaning that a lot of transcription is not productive and only some. My bigger, let's say, more a bit of philosophical question is that why would cell waste so much of energy on producing noise, for example, transcription? And it seems counterintuitive when I think about it. And does the current technology of single cell transcriptomics or single molecule transcriptomics can answer or at least get to this question? And is it really noise, or is it something that we haven't even looked at? This is not exactly health related, but this is more like a biological question I've been thinking about.
Anybody who wants to get a go about why there's noise, or whether there is noise, or just we haven't figured out what that means, or don't have the resolution to determine what it is?
I mean, I think that part of the noise, I'm not even a biologist, so please, but I would say potentially part of the noise is also like an adaptation mechanism of the cells to be able to respond faster, to be able to respond differently. So I'm quite convinced that what we now call noise, because we do not understand what it is, may have an adaptation reason for being there. Because as you say, a cell uses a lot of energy. So if we think about selection, cells that would use too much useless energy without having any phenotypic gain through it, adaptive gain, they would probably be kicked out after a while. So if it indeed uses some energy, it must have a reason for being there. Now, how to go single cell? Well, what I know, for example, at the single cell level, people have been studying quite a lot about bursting, expression bursts, that if we look at populations, we just kind of see a mean of more or less expression. Now we can really look at the individual cells and their bursting. We can look at how the two alleles, the two chromosomes, paternal and maternal, are expressed potentially differently, how the bursting may happen differently in the two. So I know that at the single cell level there is some potential for studying this problem, but honestly I don't know how much really has been done. Maybe some of you know more about it.
I think I'm just speculating here that some systems you want to have the noise and some systems you don't want to have it. Looking at the heart or the lung, for example, you want to be very precise to allow the organ to function correctly. But then looking at the brain or the immune system, you want to generate in the end plasticity to be able to go with the brain and kind of the way we are thinking in many directions, or the immune system to defend all the kind of wealth of different pathogens. So maybe here such a noise is allowing us to be more flexible and to be defending ourselves better, or thinking more out of the box. And this is maybe driven by cells and actually these noisy profiles of cells, allowing them to wobble around a little bit.
Great. There was one more question here, and then I'll go back to asking a question for the panel.
No.
But he's going to ask the question in Spanish. You might want to use this.
Oh, yeah.
Or I can translate.
You can ask in Spanish. I wanted to ask Maria, in your presentation I've seen how you use the methylome. I think we receive so much information on that is difficult to assimilate everything, the methylome. My question was the following. Haven't you been curious, or haven't you thought about, instead of analyzing everything that is expressed in chromatin, what part of the genome is never expressed in any type of cell? They are always methylated. Are they always methylated? Could you tell us this is part of our paleogenome, or if there's some other author that has already done that? What could you say at this respect?
Sorry, yes.
Yeah, the question is about the part of the genome that is silent in a more permanent basis.
Yeah.
And whether we've studied it.
I answer in English.
Puedes contestar en castellano y se lo doy.
A ver, yo creo que
I believe that the part of the genome that is silent, always silent, in silence, it's hard for me to understand up to what extent it can be functional. What may happen is that parts of the genome that should be silenced always sometimes can be not silenced, and we see that it happens sometimes. And there are parts of the genome that we know are silenced always, and normally the problem that we have is that these are parts of the genome that are very repetitive throughout their base steps, and it's difficult to measure them because as they were very repetitive, we measure them, but we see 80, 80, 80, 80. We don't know where to map base pairs. Sorry, we don't know. It was so repetitive. We don't know where it goes. Part of the genome that are somewhat dark. Now we have more and more versions of the human genome with these pairs that are better sequenced and identified. But the parts that all were silenced are parts that are very difficult to study. What we have seen with one of my collaborators is that there are parts of the genome that are not expressed, that at the level of the single-cell transcriptomics, we never look at them. But what we have seen is that what we can see is the methylome, or the opening of these parts of the genome, and using as a proxy if those parts are more active or less active. We can't measure them through the transcriptomics, but we can measure them if they are more or less active. For instance, the ribosome, the genes of the ribosome, or tRNA. Part of the genome that we know are important for the metabolism, if a cell is producing very much or producing very little, if it's replicating more or not. And we can't access that through the single-cell transcriptomics, but yes, as a proxy through the single-cell epigenome. So this idea to go to zones of the genome that we normally don't look at, but that could be reactivated and be important for a special status of a cell. But normally, these are parts of the genome that are very difficult to explore. So they have this strange other thing that makes it more complicated.
The genome activated to kind of define a cellular function with the different gene activities. But also there's a huge part of the human genome occupied by viral integration over the last, the way that the genomes or the human genome and genomes before evolved. So it is in the end, it's favorable to have sites deactivated because these are activities that we need for our functioning. But I think it's also the protection of the methylation and kind of closed chromatin, which is a silencing which can be unfavorable for us or even dangerous when those sites get active. And this is what we see in cancer and what in the end the epigenome might have co-evolved for, favoring activity but also suppressing what can harm us.
So I'd like to end the debate. We are a little bit over, but I'd like just one last reflection from each of you, if I may. I mean, we have a phenomenal resource, and you guys have a view of that resource that is quite unprecedented. What would you like to see in terms of advance for your fields using this incredible resource that we now have? 3 years from now, you're looking back. This is what you wish this human cell atlas has delivered in your field of research, very briefly.
Yeah, I mean, in terms of technological development, for me, what is very exciting is to look at things on 3D, because in a way, we are 3D, and I think that will tell us, in the end, for example, all the stem cells in tissues, they form 3D structures, and that's how you can really study them. And with the 2D, it's really limited of what we can see, and that's something that I'm excited in terms of technological development.
Yeah, I think I have two wishes. One we mentioned already is that we can agree on a way to name things, because now we have this catalog of cells and maps, and in the end, an achievement should be that we, in 3 years from now, we're speaking a common language. We name things the same way so we can talk more clearly what we actually mean when we talk about complexity and shifts in diseases. I think something that hasn't been achieved so far and something we're kind of pushing today is the transfer of single cell but also spatial methods towards clinical application. So I think there's 0 single cell application that really made it to the clinic. Now kind of also companies are pushing for that, but there's kind of this switch, this translation that is still missing today. It's probably more favorable for spatial genomics applications because it's closer to the way pathology is done already, using kind of slides and looking at tissues. So we just augment another layer, which is more a digitalized version of tissues and tumors, for example. But I think this is something we can expect happening in the next 3 years, kind of agreeing on something and talk about the same thing at the same time, but also having a translation which then eventually can make an impact for patients.
Absolutely.
So I think, to answer your question, I would say, what is this data set? And at the end, it's a resource, right? It's not a discovery in itself. So what I don't know, what I would dream about is to make more discoveries. That's what we need. And what we know from the past is that whenever you say, well, what was this discovery? Let's take a look at this discovery that just happened. Where did it come from? What we observe is that the answer, like the discovery, was right there under our nose for many years, and we just didn't notice it. And so, this resource, there's so many discoveries that are right there. And you might say, well, why don't the scientists just go in and pull them out? Why? And it's very difficult to make a discovery. It's really hard. I think we're very biased by the operating hypotheses that we have that condition us against seeing what's right under our nose. And it's not easy, and it's going to require a lot of time and a lot of efforts, and there's a lot of work to do. So I think, if we say that now we have completed this atlas, I would say, okay, now the exploration begins for the discoveries.
Yeah, that's very nice. So I come more from the epigenomics field, and obviously what I wish is that we measure more epigenome. So when single-cell epigenomics started, it started with single-cell methylation. And I think the methylome, it's a very important regulator, but it's very difficult to measure. So then when we got single-cell ataxic, which is this open and closed chromatin, everybody just did single-cell ataxic. And we somehow forgot a little bit that there used to be a thing called methylation, that used to be important back then, and we somehow don't measure it anymore. So from my more mechanistic point of view, to understand how the genome is regulated, what I miss is that we reintegrate DNA methylation as a player that we kind of forgot a little bit about, that we develop more single-cell methods that can incorporate methylation, and that we get better, less sparse, and higher quality combination of all of these methodologies in one individual single cell, because we have all of these multi-omic techniques, but there is always one of the modalities that has relatively low quality. So at the end of the day, it's not that easy to make new discoveries from this type of data. But as you say, making a new discovery, it's much more than having just the right data. Yeah. But in this case, I'm really missing the DNA methylation field that I would say has been very much neglected at the single cell level.
Muy bien, con esto.
Well, with that, I would like to thank once again, first of all, the Foundation for organizing this debate and these possible presentations. I also would like to thank all of you for your time and curiosity, and of course, the 4 fantastic speakers that have been with us here. A round of applause for them. Thank you very much to all of you.